All right, so this video is going to be about the chapter investigation for anybody who wants to do that one. I know that most people don't have a microscope and fewer people would have prepared slides of onion root tips doing mitosis. So I'll walk you through this. The first step that you want to do is make a data table like the one shown on the sample of your notebook. And you want your notebook to look like this right here. All right, so step 2 is going to be to obtain a prepared slide of an onion root tip. Place the slide on the microscope stage using low power objective. Adjust the focus until the root tip is clear. So this is what we're going to be using right here. This is a onion root tip, and if I put it on the white background, you can kind of see the onion root tips stained there on the slide. All right, the next part in stages of the cell cycle investigation, step 3. Move your slide until you're looking at a region just above the root tip. That would look like that right there, just above the root tip. The cells in this area were in the process of mitosis when the slide was made. So then look at the box-like cells arranged in rows. The DNA in these cells has been stained, and that's why the chromosomes show up. Select a cell in interphase, switch to high power, and sketch that in your notebook. Let's take a look at that then. Okay, so the next step we're going to do is step 5. In step 5, you're going to repeat what you did for step 3 and 4. Instead of looking for interphase, though, you're going to be looking for prophase, metaphase, and anaphase and telophase. And you want to reference what is on page 83. On page 83, you have examples of an animal cell in interphase with its diagram. And then the cell in the next stages of mitosis. So you have a good example of what it looks like in real life and a good example of what it looks like in a kind of cleaner diagram. The only thing I'll say is that we're looking at an onion root tip. That's a plant cell, not an animal cell. Animal cells will pinch together like this from the outside. Plant cells will actually form a cell wall starting on the inside and then working its way up. So in the next slides, I'm going to stop talking again. In the next slides, look for cells that are in each stage of mitosis. We'll go back to your chapter investigation, and then you want to sketch those stages and then kind of put them in order. So try to find things that are in prophase. What does that? Draw a picture of what that looks like. A metaphase, anaphase, and telophase. Okay. let's wrap up our investigation. So we have one more part. It's step 7. It says under low power, but I'll let you guys just go back to any part of the video that was looking at the onion root tip. Just pause it and then decide which stage the cells you see are in, and then try to record a number of cells here. So in this box right here, you want to be recording how many cells are in interphase, how many cells are in prophase, and make dashes or write down a number that you see there. And all of this, for the thing to wrap it up, would be the questions. I'm really interested in this challenge question that looks at that part. If a cell was alive for 24 hours, about how much of that time would it be spending in interphase or in prophase? And then the other one I'm really interested in is this identifying limits. So do all 6, but I'm really looking for, what made it difficult to identify those in the practical quiz yesterday? And then about how much time do these cells spend in these stages during the course of their life? Once you're done with those 6, you can take a picture of your notebook, or you could have put this on a Google Doc, attach that picture or Google Doc to your assignment, and you're all done with week 26. So attach that to week 26 and click turn in, and good job. You're done with the chapter 6 or chapter investigation for chapter 3, and you're all done with week 26. Good job.